Okay, now we're recording. Okay, this is my project. This is my current video on some Wikipedia. This is about doctors who lie. Okay, so here we are. We are going to type in neurochemistry. Okay, here's a study of neurochemistry. They have neuropeptides, neurotransmitters, the physiology of the nervous system. Okay, well, since we know this one thing about cells, cells actually have similar organelle processes. However, they differentiate enough to say that cells are distinguishably different. Okay, so that changes the pathology and the etiology and the medical therapy. The biochemistry is different for each type of cell that we have. It's not exactly the same in the products and the byproducts for both input, output, and anything in between is different. Okay, so in here it says neurochemistry is recognized science is relatively new. The idea behind neurochemistry has been around since the 18th century, which is the 1700s and beyond. Originally, the brain has been thought to be separated entirely apart from the peripheral nervous system. But it's absolutely not. It's connected. Beginning in 1856, there was a string of research that refuted the idea. The chemical makeup of the brain was nearly identical to the makeup of the peripheral nervous system. That goes into saying that different cells have very similar organelles and some similar biochemistry. So basically, let's check on our recording here. Let's do we want a little scene collection. What was I doing before? Uh, okay, well, I can't see what I'm recording. It says my camera is on. Not sure why. But, okay, we'll watch the video and see if it's good enough. But, anyway, here we are. The Neurochemistry, American Society for Neurochemistry. Uh, there's an International Society for Neurochemistry, too, and the American Society. The early gathers discussed tentative nature of possible neurotransmitter substances such as acetylcholine, histamine, substance P, and serotonin. By 1972, ideas were more concrete. Okay, they're not fully developed still yet today. There are still researchers working on this. Okay, and these doctors have to use a very complex lab to understand the neurogenesis, neuroimmunology, molecular neuroscience, list of neurochemicals, neuromodulation, neuropharmacology, neuroplasticity, synaptic plasticity, signal transduction, and overall deformation of the system and alternatives. Okay? And basically, this is a really short thing here. Uh, so, you need to know that they probably have mapped out the organelles to some extent, depending on the fact that organelles can vary somewhat based on their input chemistry and their output chemistry, and they're not probably exactly the same, and that has to do with different people too, and genetics, and how we do our chemistry, and how we eat, and what goes in and out. So things are very quantum in biological chemistry, especially when it deals in not the physiology but the medical therapy. Okay, that's where it gets even more complicated, twice as complicated, if not 10 times as complicated, to really do medical therapy. It is very so much a quantum physical science. I've heard people tell me that medicine wasn't quantum physical. You know, they act like everything's just simplistic. But we're discovering now that people have very much uh, differences, and we are selective. And biology, it can be somewhat of a selective process. And the natural selection of both being born and DNA, and even just how chemistry reacts with cells. So it's really a broad spectrum natural selection. But however, it's not so selective that it can't actually select alternatives. So that's the problem, is that we're not intelligent enough to avoid the alternatives that might be harmful. Our cells are not that complex in many of us. Maybe some people they are, but like poisons generally show true. Like if you drink a bottle of alcohol, you're drunk. So there is some established ideal science, but like the medical therapy really is quite complex quantum physically. It pertains to the energy, the physiological quantum physics entirely. I mean, and then the pathology of what could happen if there is an alternative to the flow and the functionality of these mechanisms we call medicine. And even in genetics, and how we age, and what's going on with our body at different times in our life, and depending on what diseases we have. It is very complex, so I want this to be known that you shall never, ever absolutely trust a doctor, but more so you need to trust yourself. Uh, you really, we all need to do medical research. It's that complex. Uh, Especially if we want to live beyond the established human uh, lifeline. The what's the word? The established human life expectancy. That's what I'm going to say. So, like right now, stem cell programming is developing pretty much. And that's a sign. Genetics and stem cells um, might be able to heal the body quite a bit. And I'm hopeful that someday it will for the future of mankind. But where are we at on that now? They're saying that they have performed successfully CRISPR. Uh, let's do this. Jennifer Doudna did some remarkable work in CRISPR. Um, okay, CRISPR is used for cell cultures. And then here's one for T cells in patients with refractory cancer demonstrates that such CRISPR-based therapies can be safe. April, June 20th in Science, successful treatment during a small trial of the first use of CRISPR gene editing. Okay, so let's start a new page. This is the one that I want to read. NPR.org. A patient hopes gene editing can help with pain of sickle cell disease. Here she is. She's a black woman, African American, I'm guessing. And she's not African American. I don't know. Victoria Gray slides open a door closet, pulls out a suitcase, and starts packing piles of clothes. Okay, so she says she's from Nashville. She's going through a clinical trial. Her doctor is Haydar Frangal. Okay, Victoria Gray. I had that confused in another video. Uh, this is her. Beautiful lady. Um, but sickle cell disease is a devastating genetic disease that affects millions of people around the world, including 100,000 in the United States. The disease is caused by a genetic defect that turns red blood cells into hard, sticky, sickle-shaped cells that don't carry oxygen well, clog the bloodstream, damage organs, and causes torturous bouts of pain. Okay, this pain is excruciating, like being in a car accident. Like kind of how my leg feels right now. Because it's numb. Because I have sciatic nerve problem. Uh, but here it is. Modify the genes in the cells to make them produce fetal hemoglobin. a protein that babies usually stop making shortly after birth. The hope is that the protein produced through the gene editing treatment will give sickle cell patients like Gray healthy red blood cells. CRISPR enables scientists to make very precise changes in DNA, raising hopes that the technique will lead to new ways to prevent and treat many diseases. In July, one of the companies revealed the first hint that the approach might be working, at least in beta thalassemia. The patient's edited cells started functioning in the bone marrow, the company reported. Doctors are also trying to use CRISPR to treat cancer. Most of those studies are underway in China, but 3 patients have been treated for cancer in CRISPR study at the University of Pennsylvania in Philadelphia. And another study recently started recruiting cancer patients in the U.S. and Australia. Later this year, doctors in Boston plan to use CRISPR to edit cells in patients' retinas in hopes of restoring vision in patients with inherited forms of blindness. Oh my God, this is so awesome! Because I've been talking about all this. I have an RT caseworker person who has blindness, and I've been in love with the whole stem cell thing and gene editing and CRISPR and all this stuff for a while. I've been studying medicine for 8 years independently, and I'm also going to college. But oh my God, this could be it. I mean, I'm praying. I am so much praying. My prayers could never end. This is a big moment for Victoria and for the pivotal trial, because if we can show that this therapy is safe and effective, it can potentially change the lives of many patients. The study is being conducted jointly by Vertex Pharmaceuticals in Boston and CRISPR Therapeutics in Cambridge, Massachusetts. 8 recruiting patients in the U.S., Canada, and Europe. Up to 45 patients aged 18 to 35 will eventually be enrolled. We're hopeful this will work for Victoria, but we don't know that yet. This is very much, this is very hopeful. Okay, this is something to follow. This is NPR.org. Afterlife of painful sickle cell disease in patient. Patient hopes of gene editing can help with pain of sickle cell disease. But that's pretty well the end of my video. And thank you for watching. And I hope that you like it.